welcome to this lab wherein we will look at how we can use COBRA Toolbox to do simple simulations and so on. We will first study this all important function called optimized CB model, and then we will look at what are the exchange reactions in a model, because it is central to our understanding of how to manipulate the medium for growth of an organism. And we will also do a trivial experiment wherein we vary the glucose uptake rate via in the model and see what is the potential effect on the growth rate. Welcome back. Let us look at how we study constraint based models using the COBRA Toolbox. So, the first thing to do with the COBRA Toolbox is you need to initialize the toolbox. So, that is assuming you have already downloaded the toolbox and added it to the path and so on. So, you say init COBRA Toolbox. So, this is important to note. It says that you can solve LP problems using GLPK, Gurobi, MATLAB, PDCO and LP solve. You can solve mixed integer using GLPK and Gurobi. GLPK is like free, right? Gurobi is free for academics. So let us try out a few things. First thing is, let us again load the E. coli core model. So, it goes into this thing called model. So, we already looked at the structure of model in the previous class. So, model looks like this. Now, you need to understand how the COBRA Toolbox works. So, let us go to the COBRA Toolbox directory. It should be on a similar folder for any of you, and as I mentioned a long while ago, help cd is useful, but there are only a few commands here. So, init, which happens to have no documentation, and update COBRA Toolbox. So, let us go inside of source. All these are available in your path, by the way, right, and if you put help cd here you will get nothing because there are no M files out here. Let us go into model analysis. Yeah. This tells you a bunch of interesting methods. So, there is something called flux enrichment analysis, there is dynamic FBA, geometric FBA, phenotype phase plane analysis, so on. But the one useful thing here is biomass precursor check, which lets you know if biomass precursors can be produced and so on. We will come back to this in a moment. I am just trying to give you a feel for the COBRA Toolbox. So, this has some basic functions for parsing and things like that. So, model analysis is a very useful folder. So, if you go inside FBA, this is the most important function by far, optimize CB model, and let us look at the help. Yeah. Yep. So, it solves the LP problems of the form max or minimize C transpose V such that SV equals B and LB less than equal to V less than equal to UB, right. And you can also give a few other things like should you allow loops and do a zero norm approximation or a minimum norm or things like that. We will look at what they are in subsequent classes, most of them. The start is basically 1, 2, 0 or minus 1 and is a translation from the solver specific status, right. So, every
solver might return its own status. So, GLPK might return 0 for a successful solution and GROB might return 1 for a successful solution, but that is translated to a sensible status here. So, this normalizes across solvers essentially, right. So, unless this holds, do not look at your solution, it is not useful. This is an important thing to remember. What are the inputs? I think you know it very well by now. The stoichiometric matrix, the right hand side usually zeros, objective coefficients, lower bound, upper bound, right. And which way do you want to optimize it? You want to maximize biomass function, or you want to minimize ATP uptake or whatever, and so on. There are interesting concepts about min, norm and so on, and I think I will dwell on this a little later. Let us keep things simple for today's first lab on the COBRA toolbox. Yeah, let us skip through most of these things. So, these are all optimization specific things. There is a primal and dual for every linear program. I will not get into those details, but you can in any good optimization course will cover all of those. Okay. So, how do you use it? Once again, you say FBA solution equals optimized CB model of model as input, right. So, the model I just loaded and this gives you the solution, right. It used the GLPK solver and took about 0.6 seconds to find out that the growth rate is 0.8739. For a real FBA problem, you would need to start worrying about the nutrients. So, essentially have a cell, there are several nutrients that come in, some nutrients that grow out, and let us say there is biomass, right. So, what are these? So, these should be mentioned by some fluxes, maybe you want to at least give glucose, right. So, you will have to see where this is, and then you have a metabolic network inside of the cell which converts between all these molecules. So, where is this nutrient specified? One way to look at it is, so I think you know by now, suppose I had a reaction like this, how would it figure in the stoichiometric matrix?
As B1.
That is it, right. So, just -1 for this, and for A it will be for B it will basically be 0 1 0 0 0 and so on, right. So, these are exchange reactions and you can easily find them in the model. So, this tells you what are all the exchange reactions in the model, and if you say find, it turns out that reactions 20 to 39 are all exchange reactions. So, let us look at what these reactions are. So, it turns out that this model can exchange acetate, acetaldehyde, alpha ketoglutarate, carbon dioxide, ethanol, formate, fructose, fumarate, glucose, glutamine, glutamate, proton, water, lactate, malate, ammonium, oxygen, phosphate, pyruvate and succinate, right. So, these are all the things that these are all the metabolites that this organism can, this core model of E. coli can exchange, right. So, now how do you figure out what is the, you may want to figure out what is the glucose, right. So, let us look at all these lower bounds and upper bounds. So, I want you to recall how we do logical indexing in MATLAB. So, this is basically a logical vector and I can just use this. So, if you see most of these have a lower bound of 0, which means that they are only expected to be secreted out, whereas and several others have a lower bound of -1000, which means they are just like arbitrary exchange fluxes. The one with minus 10 seems to be the most interesting, right, because that sounds to be like a specific constraint that is given, everything else is like default, 0 and minus thousands are like default values. Find exchange reactions. We look at upper bounds, it is all 1000, no surprises there. So, now what is this minus 10? So, let us just find mod dot LB equals minus 10. Hopefully, there are not others, there is only one which is 28. Total model dot reactions 28 is glucose exchange, right. So, now let us double the glucose exchange rate, right. So, now let us say model dot LB 28 equals minus 20. Now, let us again grow the cell, you find that the growth rate is almost doubled, right. So, you see that the growth rate keeps increasing, let us see if it keeps increasing. I am like, now it only doubled. So, it's something else has become limiting, and that is very likely to be oxygen. So, can you try out a few things now? Are you able to first do this FBA solution? It is trivial, right. So, this works. What is the objective function in this case? How would you find out the objective function? So, how do you know that that is the objective function? It is actually embedded in the model, right. So, it is going to be in model dot C. So, model dot C has obviously a lot of zeros. So, let us say find model dot C. Turns out that the 13th reaction is being optimized. Let us find out what is this reaction. Oh yes, it seems to be the biomass E. coli core model with growth associated maintenance. We will see what that is in a later class, but for now let us look at how this reaction looks like. There is thankfully a piece of code in COBRA Toolbox called print Rxn formula. So, if we say print Rxn formula of model dot reactions 13, it will give you the actual biomass equation. So, the biomass equation looks like this, we have not seen it before. So, it makes sense to see it in for a simple model such as this. So, it says 1.5 moles of 3-phosphoglycerate plus 3.75 moles of acetyl CoA plus 59 moles of ATP whatever gives 59 moles of ADP, alpha ketoglutarate, CoA and proton and NADH and so on, right. So, this is the fictitious reaction that is being optimized. What this means is you need 3PG, acetyl CoA, ATP, erythrose 4 phosphate, fructose 6 phosphate, G3P and so on in this ratio, right, for the cell to grow. If this is not available, I mean, so this will be how the model limits the growth of the cell. If you suddenly have very low F6P, it is going to substantially lower the growth rate and so on.
How do we, where is the exchange reaction is stored in the model and the reactions metabolites?
It is only in LB and UB. The exchange reactions are stored only in LB and UB.
LB and UB are just upper and lower bounds, right?
Yes. Okay. So, if you want, you can also look at the corresponding stoichiometric matrix. So, model dot S colon comma 28 should tell you the exchange reaction for glucose. That is only -1 0, exactly like this, right. So, let us look at what is 35, model dot METS 35, glucose external, right, model dot MET names 35, sorry 35 D-glucose, right. So, this D-glucose coming into the cell, that is reaction number 28, and all your exchange reactions are like that. So, let us just do that, no. You find model dot These are all your exchange reactions. If you now say model dot S colon comma this, that's it. Yeah.
So there is nothing going on.
Well, some of these have reversible upper bounds, minus 1000 to plus 1000, so they can also go out.
Okay.
You have to look at these numbers in the context of the LBUVs as well. As far as the mass balance is concerned, you will be only able to, unless you had measurements for a reaction, I would only be able to tell the net flux. I would say 75 plus 75 minus 50 equals plus 25, but it could very well be plus 175 minus 150 equals 25. Right? You have so much variation available there, and if you actually try to optimize for a sparse solution, you will end up getting 25-0. No, there is no way to find out, there is no way to resolve these internal fluxes unless you have some 13C labeling and so on. It's hard. That's a very good question. So in fact, we will see when we talk about elementary modes that this is one way to represent reversible reactions. You can show them as two separate reactions. It's useful for finding extreme pathways and so on, because then all reactions are irreversible, right? If you put them as two irreversible reactions, yeah. So we studied a few concepts here. We looked at find exchange reactions and we also looked at print Rxn formula. So let's say you want to change the growth substrate now. How do you change it? Suppose I want to see how E. coli grows on acetate.
You want to substitute or add a
I want to substitute glucose with acetate.
change that row?
So I would basically need to find out where is acetate.
And make it
So model dot Rxns 21 to 40. Maybe acetate is 20 already. Yeah, acetate is 20, right? So it makes life easy. So we just say model dot LB 28 equals 0. This means there are no exchange fluxes now, and obviously you can check that the model does not grow. It's basically inconsistent even, so your status itself is 0, right? So there's no glucose, so it does not grow. So if you make it a very small amount of glucose, you will find some very slow growth. But let's remove glucose and add acetate, and so we find that the growth is 0.1733, whereas the growth on glucose was 0.8739, right. So we can check that again, model dot LB 28 equals minus 10, FBA solution equals optimize CB model of model. Yeah. So 0.8739 versus 0.1733.
So initially acetate was 0 then.
Initially acetate was 0. Initially everything was 0. So it was like this, model dot LB 21 20 to 40.
There are minus 1000 also.
Yeah. So but those are different things. So this is 20, 21, 22, 23. So model dot that's carbon dioxide.
And sir, if we make it 0, there is no incoming or outgoing. Is that the meaning?
No, it's just the LB. So it means that there is no incoming. It depends upon the direction of the reaction.
So if we make plus 1000 in LB, so what does that mean?
Which means that you have to secrete at least that much of. It depends upon the reaction. So to answer your question, so let's look at model dot. Let's say print Rxn formula reaction 28, right. This means glucose coming into the cell. If you say plus 1000 for this, it means that 1000 glucose has to go out of the cell. So if you say minus 10, it means 10 glucose is uptaken. If you say plus 1000, it means 1000 glucose has to go out, and that might never work. The value of this reaction is at least this much.
Suppose you want to change how much acetate. So first you have like a nutrient medium, all the nutrients are there, and then you change the ratio of the nutrients. How would you do that in the model?
So now let us say I want to add some acetate. So I will say model.lb20 equals minus 10. So now I have 10 of glucose and 10 of acetate. So if you say I have 10 of acetate and 10 of glucose.
So LB is like an initial concentration.
LB is the uptake rate.
Yeah.
There is no concentrations, remember.
Huh.
In flux balance there is no concentrations really.
That is the minimum you want so that the reaction will take place.
So if you want to represent concentrations of nutrients outside, you have to do it as exchange rates.
Yes.
And
Yes.
represent it
Yes.
as
It's not concentration, it's uptake rates. We are talking about a steady state system.
Sir, so
Right?
why you find
So there is 10 millimole per liter per hour of glucose being uptaken by the cell.
is growing cells. All she knows is how much she's adding. Like how do you?
This is a steady state. So you're thinking you should not compare with something that's grown on a Petri plate. You should think of something that's being grown in a reactor, right?
Okay,
A
in a
CSTR.
reactor.
So in a CSTR, you know what is the initial concentration of glucose, you know the flow rate, and you know the final concentration. So you can compute the steady state uptake of glucose by the cells, right?
Oh,
So this is that input.
so you can't perform experiments on like small cells.
Yeah, so this is a steady state modeling thing. So if you want to generate data for this, you will have to set up a CSTR kind of setup.
Sir,
This is stirred reactor. Yeah.
the answer is giving the LB of all exchange reactions.
Yeah.
So why there is a 0 there? For some
So maybe they should be segregated. So
Ah.
maybe we should just have a simple.
so we should check UB also.
LV coming.
So whichever are 0, if something comes in for those metabolites also, then the cell won't work, the cell won't grow or work. So, like if something has 0 and but the metabolite is coming inside, so then the cell won't.
What do you mean the metabolite is coming inside?
In the exchange reaction, a lower bound is 0. So nothing should come inside.
Yeah.
So if something comes inside, it means that the cell won't grow or.
No, I mean this is a model, right? So this is modeling the condition where nothing is coming inside.
Okay.
The 0 means that nothing is coming inside,
Okay.
right? So this is trying to capture what happens in.
Okay.
The vector V is supposed to have reaction rates and an exchange rates.
All the rates.
All the rates and.
Right. So if you remember in our example, we said we will split it as V1, V2, B1, B2, B3 and so on.
Convention or general?
It's just convention. It's just for teaching, not even convention.
So like
So now it's all just mixed up,
now it's all.
which is why you need a code called find exchange reactions and print exchange reactions. So I'm just trying to write a small piece of code that will print the exchange reactions in a sensible way, right? So
will be same fluxes. Yeah, this flux means AH lambda flux, BH lambda. I think so. I think I'm not sure. Type in COBRA Toolbox. You do. Single toolbox. Where are you? Are you in? Do it again and check for
Okay. So, now we have a small piece of code that tells you what are the exchanges and how do they look. I just wrote a new piece of code which says print exchange reactions. It says okay. So, this is acetate both ways. So, minus 10 means
minus 10 and come in.
10 comes in. So, this is how much glutamine is secreted, this is glutamate is being secreted, ethanol and so on. And ammonium, oxygen, phosphate, right. So, maybe let us try out a few things.
All those forward reactions, LB is 0.
Yeah,
Means
they are anyway irreversible, right. So, you see the arrows that is shown here, all these are irreversible.
yeah, so they will only come out. Okay. Oh, the other things can come in and go out.
Yeah.
So the biomass reaction, it had all the nutrients or reactants in certain ratios.
Yeah.
So if you change the LBs or whatever corresponding UBs of those fluxes, such that, so say you have 5 nutrients in the ratio 1 to 1 1 1. If you increase one of those such that it doesn't, if you increase the concentration of the excess reactant and then you run it, it shouldn't change the final answer.
Yeah, the limiting
If you
reactant will be
increase
same.
the LB of an excess reactant,
Yes, yes,
it
it shouldn't change, right. And in fact, here you will find that, right. So, here let us say let us actually do that for glucose uptake rates equals minus 10 minus 2000 colon 10 colon minus 10, right. Let us say model dot let us just or let us make a few more, just make a few steps. So, minus 2000 minus 1800 minus 1500 minus 1200 minus 1000 minus 800 minus 500 minus 200 minus 100 minus 50 minus 10 minus 10, right. So, and let us say empty, let us not worry about writing efficient code and let us just put this up. It model dot LB 28 equals GUR FBA solution equals model dot equals optimize CB model of model, right, and GR rate equals GR rate FBA solution dot X, right. Now, plot GR rate
Y dot x.
the X value, because FBA solution is a structure.
Yeah.
plot sorry, I made a mistake. It should not be x, it should be f. That is what you were trying to say? Yeah. So, let us say GLC rates equals this, right. So, for GUR equals GLC rates. Now, let us also initialize growth rate to empty. For GUR equals GLC rates, model dot LB is GOR, if FBA solution is optimized CB of model, GR rate equals dot F. And now we basically can plot GLC rates, GR growth rate. In fact, I'd like to plot it this way, right. You want the magnitude, right. So, if you keep increasing, so at this point it flattens out.
Whatever you increase, it is just excess and it's not getting.
Yeah.
But sir, so by that logic, if we increase a limiting reactance LB, then the answer should change.
Yes.
It should increase.
So now if I did this for a different oxygen rate, so let's just do this model dot, let's do print, where is oxygen? This must be 40, 39, 38, 37, model dot RXNs 37, 36. So, model dot LB 36 is equals minus 1000, let us make it minus 100, right. And now let us just repeat this. So, I have a new set of growth rates, I will just say hold on and plot it again. I did not reset the value. So, this should be size GR rate 22. So, I need to plot So, I reduce the glucose uptake rate, the oxygen uptake rate.
Lower bound you.
Yeah. So, I made oxygen as 100. I reduced it by 10-fold.
So minus 100 means you are increasing the uptake rate, right?
No, from it was minus 1000 initially.
Oh, minus 1000.
Oxygen was minus 1000, glucose was minus 10, and I am sampling glucose at all these points. So, let us now you know the exact points where we did it.
why should it change though? So if you have say two constraints and one objective function and it intersects at one point, that is the answer in the first case.
Model dot FBA solution dot x 28. What should this value be? Wait, we don't even, let's first check what is model dot x 28 because we've been playing around with the numbers. Oh sorry, model dot LB 28 is minus 10, right? Model dot LB 36 is minus 100. What is FBA solution dot LB 28, sorry, dot x 28 minus 10? FBA solution dot x 36, that is the lower bound, right? It can take it for 10, it needs only this much oxygen. But if you now say model dot LB 20,
So now if you change 36,
hold on, model dot LB 28 equals minus 500, okay? And now let us say FBA solution is optimized CB model. Now you say model dot LB, model dot, sorry, now you look at FBA solution dot x 28 it's minus 500, x 36 is going to be a scaled version of this, the previous number that we saw. It's actually limited at 100.
Now oxygen becomes limiting.
so depending on the ratios, you can't say that it will for sure change, no? Because if you have constraints.
Yeah, see, you have to look at all the reactions. So you need to now look at how, see, the rest of the glucose is probably just going somewhere else, or it's I don't know if there's something else that's dumping glucose back and so on, right? If there is some cycle where glucose is consumed, because you're clearly limited by the oxygen you have, right? So maybe I don't see know why the cell is actually taking up 500 of glucose now. 500 was the LB we gave it, but that needn't be the glucose uptake rate simply because it's going to be oxygen limited now.
But since we are optimizing at glucose,
We are not optimizing at glucose or anything, we are optimizing for biomass.
C was 28, right? C was 128.
No, C was 13. 13, right? And model dot Rxn names 13. So essentially, this was a brief intro to how you play around with you know, the uptake rates and find out where the objective function is and make a very simple, you know, analysis of this sort and so on. So we will incrementally look at much better applications of FBA. We will try to do some more applications of FBA in the afternoon and we will build from there on. No, afternoon I'll give a class. So next week we can do some nice labs.
Sir, then what is the reason it is taking 500 glucose, minus 500 glucose?
That's interesting, I'm not sure. So we'll have to see. So it's not enough if you just compare the x value of glucose, you need to look at the entire x vector. Where does this x vector deviate from the older x vector?
Okay, so because if you have like your original model two constraints and your objective function intersecting, and then when you change a lower bound, you're not changing any of the functions, right? So if that
No. So that one line or one constraint was actually one lower bound, right? It's an inequality constraint on the model. You are saying instead of
horizontal or
no, it's
vertical constraint, no. If you have like two.
so you had something like this. Instead of exploring in this area, you are now saying explore in this entire area.
Right.
right, because you lowered one flux, right. So, LB went from minus 10 to minus 20. So, you opened up a larger area for exploration.
But if you make it tighter, that's what, if you decrease the lower bound, the magnitude of the lower bound, and such that it doesn't cross.
So
the
then
lower
it
bound,
it
then
it,
it
it
didn't change,
no, you get new corner points, which may be optima. Let me draw it better. Let us say these were the optimal points you were looking at before. Now you get another set of places where the line cuts the feasible. So, you have two more optima to explain. It may change, it may not change, you can't guarantee, but I really like the fact that you are thinking from the geometric perspective.
So, in this lab I hope you got a good picture of how we can perform flux balance analysis using the COBRA toolbox and identify the exchange reactions, change the medium of growth, add multiple substrates and so on. And we also did a simple experiment wherein we varied the glucose uptake rates and saw the effect on the growth rate of the organism. So, in the next video we will look at some very central concepts to understanding flux balance analysis, particularly how do you troubleshoot a flux balance analysis problem wherein you have a model but it often does not show growth. So, we will first look at the concept of gaps, then the concept of dead end metabolites and blocked reactions.